So we would like to welcome all of you joining us across the world in a new global IMMUNOTALK. But before we start, we would like to share with you some very important thoughts. So these have been very, very difficult times, and we strongly agree with the principles of shut down academia and shut down STEM. And we have carefully thought about the best way in which we could support this movement today. We are deeply aware and condemned every injustice and all inequality everywhere in the world. And we truly think that this requires systemic efforts to improve access and opportunities to all of us. And it is in this sense that we think global IMMUNOTALKs provide a clear example of concrete actions. We think that it is in this way that we can try to improve access to education and knowledge to all of us in an inclusive manner worldwide. So with this in mind, we decided that the best way to support the sentiment of our colleagues that today are shutting down academia and shutting down STEM, continuing with our mission, which is to have this talk. We truly hope that our colleagues understand our decision. And just as a reminder, as we do in every global IMMUNOTALK, we would like to share with you what are the main goals that have really driven our actions in this regard. And that is to benefit and inspire immunologists everywhere in the world in an egalitarian manner. This is why we have developed these talks that are free, that are uploaded in YouTube, so you can watch them later on. And we also have a virtual discussion that you can have later on, today or tomorrow. And we also think that through these global IMMUNOTALKs, we can increase opportunities for scientific learning without traveling. And traveling is many times something that limits our inclusion in this scientific world. So with that, I'd like to introduce Elina Zuniga, my friend and colleague, who would introduce Kate Fitzgerald, our speaker today. Thank you, everybody.
Thank you, Carly. So I couldn't agree more with Carla's word. I want to make as clear and state as strongly as possible that we repel any type of discrimination, economical, social, and of course, racial discrimination, not only in US, but everywhere in the world. And the humble, concrete action that we have been taking to fight that discrimination has been to implement these global IMMUNOTALKs, and with the hope that they make the world a little bit more inclusive and egalitarian. And so that's the decision, or the basis for the decision we took today. So with that, I would like to introduce our amazing speaker today, Dr. Kate Fitzgerald. Kate is originally from Ireland, and as I heard, she's a very proud Irish. And so Kate has received her bachelor degree from the University College Cork in Ireland, and her PhD degree in biochemistry from the Trinity College in Dublin. She then moved to the States to perform a postdoctoral training at UMass. And in 2004, she was named Assistant Professor of Medicine in UMass, and then escalated quickly through the ranks, becoming Professor, Director of the program in Innate Immunity, and currently she's the Vice Chair of the Department of Medicine in UMass. Kate is also the president of the International Cytokine and Interferon Society, and we thank her very deeply for that important service. So as you know, Kate has made a seminal contributions, fundamental contributions to our understanding of how viruses are detected and how that detection then is transduced via signaling to induce antiviral immune responses. She has also contributed to understanding how the immune responses are regulated to, on one hand, clear an infection, but at the same time, avoid excessive inflammation that can lead to chronic inflammatory diseases and autoimmunity. So her work has many times significantly pushed forward the field of innate immunity, and she is highly recognized and respected by all of us for those contributions. So this is reflected in many seminal publications that she has listed in her CV and that have been highly cited. She has been among the top 1% most cited scientists for work in immunology from 2014 to 2019. She has a long list of awards, very difficult to go through all of them, but in 2020, I'd like to mention she has been elected member of the Royal Irish Academy and also elected fellow of the American Society of Microbiology. So, Kate, thank you so much for being here today and for all the effort that implies preparing for these talks. We are deeply grateful.
Great, thank you so much. Yeah, thanks so much, Alina and Carla. And I want to thank you both for doing this. I think this is an incredible service to the immunology community around the world. And I know here at UMass, everyone trains in religiously weekly. So it's really been absolutely fantastic. And I also want to echo what you said, supporting the movements today. I think we as a scientific community have to embrace diversity. I think we do, but we need to do better and continue to encourage, inclusivity and bring as diverse collection of scientists together at the highest levels of our enterprise. So thank you.
Thank you, Kate. So always what we do before starting the scientific seminar, we ask the speakers a mentorship question that we hope can be helpful to our young audience and give them a little flavor of the amazing mentors and role models that we have among the speakers in the Global IMMUNOTALK. And as we know, not everyone has easy access to role models, and we hope that via these questions, we can fill in a little bit that gap. And so talking about role models, the question we would like to ask you, Kate, today is, who is or has been a scientific role model for you and why?
Yeah, so I think I've been fortunate to have had many fantastic mentors and role models over the year, but I think someone who really stands out was Paula Pither-Rowe, who many of you may know. So Paula was a Czech scientist. She was professor of oncology at Johns Hopkins, and I met Paula actually when I was really a very sort of junior postdoc. Actually, I think the first week I came to the US, and she was sort of a mentor to me really throughout my career, until she sadly passed away a couple of years ago. But she was just so incredibly generous and supportive, and she opened her freezers to me. This was sort of back when, TLRs, all we knew was they activated NF-kappa B, and of course Paula was the queen of interferon biology and the IRFs, and she sort of opened her freezer and said, here, take every one of these IRF plasmids, see if they're involved. So we started to chat, and yeah, just that generosity I think is so critical, and I try hard to sort of emulate that myself. So let's see. I think I do a good job, but
I'm sure you
do better.
I'm sure your mentees agree with that, Anna, and yeah, how important is the role models and the people we cross during our careers. Yeah, how important.
Yeah. Okay, so
yes, you can
share my screen and begin.
And yes, we're gonna wait.
Let's see. Desktop, I think, works best. Okay.
Perfect.
See that okay?
There
we
you
go.
go. Yes,
Okay.
excellent. Thank you, Kate.
All right, and I think I'm going to turn off my video just while I present.
You don't have to.
Okay, all right.
Yeah,
Okay.
you can leave it on, yeah.
There's nothing I love more than to see my own mug up in the corner of the screen, but nonetheless, okay. All right, so today I'm going to talk about sort of an overview of some of the work in my lab over the last several years, really focused on the innate immune response, and particularly on nucleic acid sensing pathways, and how, you know, the identification, discovery, characterization of these pathways, both within my own lab, but by numerous others across the globe, have really sort of helped us understand both the beneficial as well as the harmful effects of recognition of nucleic acids. So just before I start, I just want to highlight, so here on my opening slides, I have a collection of flags. So these are, you know, students, postdocs have come from really all over the world, and, you know, in the global IMMUNOTALKs tradition, I think it's absolutely fantastic to have this diverse collection of people. Okay. Oop. All right, so thinking about the innate immune response. So we know the immune response is really sort of comprised of two key components. So the innate immune response and the adaptive immune response. And, you know, the innate immune response is comprised of many different types of cells. I mean, all cells are really innate immune cells, but some are just professionals, like macrophages, dendritic cells. But you know, even thinking about epithelial cells, for example, you know, they recognize and respond, for example, to viral infection. And of course, when the innate immune response is able to recognize microbes through pathogen-associated molecular patterns, and in addition, can respond to self molecules. And for the purpose of today's talk, we'll really focus on DNA and RNA sensing molecules, and these different cell types of the innate immune system are really kind of decorated with different combinations of receptors that can distinguish these different types of molecules. And then the innate immune system not only directly senses microbes and elicits direct antimicrobial activity, but also, of course, instructs the adaptive immune system. So if you think about the timing of infection, the innate response is really a rapidly mobilized, happens within minutes to hours to sort of days post detection of foreign entities. And then this leads to activation of the adaptive immune system with involvement of lymphocytes in particular. And of course, you know, these early innate responses are critical to help instruct the adaptive immune response. So my lab, really, since I've had my own lab for almost 20 years now, we've really been focused on understanding the molecular basis of inflammation, both in an acute setting as well as in the context of chronic inflammation. And when we have an innate immune response, so these sort of early responses to infection, there's a rapid mobilization of sort of this inflammatory state. This can help to clear pathogens directly, or certainly curb pathogen spread. And then over time, this response, this inflammatory response, will usually resolve and sort of return back to homeostasis with a resolution state. And of course, in many human diseases, shown here with several examples with atherosclerosis, neurodegenerative diseases, arthritis, etc., that sort of set point never returns to baseline, and you end up with a chronic state of inflammation. So for many years, we've really been sort of focused on understanding what are these early responses that drive these protective defenses. And then over the last probably, 10 years or so, we've really been focused on how kind of dysregulation of these normally protective innate responses can lead to chronic inflammation and contribute to a myriad of inflammatory diseases. So I'm going to focus today on nucleic acid sensing. In my lab, we do a lot of work on both nucleic acid sensing pathways as well as inflammasome pathways. But we know now from work of many in the field that one of the main mechanisms of sensing pathogens is through detection of nucleic acids. So viruses, RNA viruses, for example, DNA viruses, even bacterial infection can be detected through mRNA, for example, DNA in the genomes of these microbes, and even cyclic dinucleotides can be released from phagocytosed bacterial pathogens, and then these molecules can also serve to activate innate immune receptors. And cells are equipped with a plethora of both RNA and DNA sensors that collectively survey cells for signs of infection, and they decorate different compartments of the cells. So the plasma membrane, the endosomal compartment, the cytosolic compartment, and even the nucleus, and detect nucleic acids from pathogens, and then turn on and mobilize these transcriptional programs that primarily lead to interferon and cytokine responses, but also cell death responses. So as I said, myeloid cells, which are really the sort of professional antigen, our professional innate cells are really a large focus of my lab. And we know these cells are decorated with different types of pattern recognition receptors that recognize all sorts of components of microbes. But in the case of nucleic acids, the endosomal TLRs can recognize RNA from viruses, for example. TLR9 can recognize CpG DNA, TLR3 double-stranded RNA. We have this pathway here in the cytosol, the cGAS pathway responding to DNA, and then in work I won't really get into today, another DNA binding protein that we worked on extensively, the AIM2 protein, can also sense DNA and turn on inflammasome pathways and control caspase 1 activation and proteolytic maturation of cytokines like IL-1. So today I'm going to really focus on these pathways that culminate in induction of type 1 interferons. So these are illustrated here. So TLRs, RLRs, and cDNA receptors, shown here with cGAS. So in response to different types of viruses, these different sensors can detect viruses through recognition of viral genomes or nucleic acids that accumulate in virus-infected cells. These then elaborate signaling pathways that engage adapter molecules, shown here. These then converge and activate kinases, a kinase called TBK1, one of our favorite molecules in my lab. And then TBK1 really serves as the kinase that phosphorylates and activates IRF3. IRF3 then turns on type 1 interferons. So in all cells, these pathways can be activated, and usually interferon beta is the initial cytokine made. And then these cytokines are produced and feed back on the same cell or on neighboring cells in autocrine or paracrine fashion to turn on transcription of a battery of interferon stimulated genes. And it's the products of these ISGs that sort of target and thwart virus infection and really shut down viral replication. So as I mentioned, my lab, we're particularly interested on DNA sensing pathways. And just to orient everyone, there's been a lot of work in the field describing many different DNA binding proteins that contribute to innate sensing of DNA. Just for the purpose of today's talk, I'm really going to focus on the cGAS pathway, but there's also TLR9, AIM2, which, as I mentioned, forms this inflammasome pathway, and the cGAS pathway. So cGAS represents the front end of what we think of as the cytosolic DNA sensing pathway. And the idea is that in response to virus infection, if nucleic acids can accumulate in the cytosol, those nucleic acids can then be recognized by sensors like cGAS. And cGAS binds DNA and it converts ATP and GTP into a second messenger called cGAMP. And this second messenger then binds and activates a protein on the ER called STING. And then STING translocates, activates the TPK1-IRF3 pathway or the NF-kappa B signaling pathway to turn on interferons, cytokines. I will mention that there's also some compelling evidence that, you know, cGAS may not be exclusively cytosolic. In some contexts, it can be nuclear, and perhaps for some of these herpes viruses, for example, the detection of DNA may actually be occurring in the nucleus rather than in the cytosol. And furthermore, there's also some evidence in some contexts that cGAS can be localized to the plasma membrane. So the main pathways that are elicited upon cGAS sensing of DNA are interferons and cytokines. But it's also worth noting that autophagy is another important antiviral mechanism that's elicited by the cGAS STING pathway. And in human cells in particular, the cGAS STING pathway has been suggested to sort of converge on the NLRP3 inflammasome pathway. So work from many in the fields have shown that many different types of pathogens are detected by cGAS, and the cGAS STING pathway plays an important role in controlling these infections. In certain contexts, it can actually benefit the pathogen itself, for example, and facilitate pathogen replication. So we've been interested in the role of different DNA sensing pathways in the detection of herpes viruses in particular, and that's shown here. So this is just taking bone marrow-derived macrophages from a variety of different mouse strains. So wild type P204 is a mouse homologue of IFI16, or at least one of the mouse counterparts to IFI16, which was proposed as an important DNA sensor. cGAS knockouts, and then the STING golden ticket mice are a functional null allele. So you can see that herpes simplex virus 1 and 2 are clearly detected through the cGAS STING pathway, and IFI204 does not contribute to this particular response. You can also just study these responses by transfecting cells with double-stranded DNA, and whether it's dendritic cells or macrophages or fibroblasts, cGAS and STING play an important role in sensing this viral pathogen. So we also wanted to address the importance of this pathway in vivo in detection of herpes simplex virus. So here we're using a McRae strain. So this is a neuroinvasive strain of herpes simplex virus. In susceptible mice, it causes herpes simplex encephalitis. You inject or infect these mice through the cornea. And then in wild-type mice, usually this virus is pretty well controlled, but in mice that are compromised or unable to control viral replication, over time the virus will spread and enter into the brainstem, and you can detect virus within the brain. And shown here, if you infect wild-type mice or cGAS knockout mice in this manner, you can see that the cGAS knockout mice are incredibly susceptible to this viral challenge. And this is true regardless of the route of infection. So that was a sort of a more physiologically relevant corneal model. This is a retroorbital model, and similarly, wild-type mice are pretty well able to control this infection. They develop very sort of minimal kind of clinical signs of herpes encephalitis. But the cGAS knockout mice, you know, lose a lot of weight and really develop high levels of encephalitis. And this is really directly related to very high levels of virus in both the trigeminal ganglion shown here and also in the brain tissue. So this clearly illustrates the importance of this pathway in sensing this virus. So just to touch on sort of some newer work in the lab. So the STING has not only in sort of academic labs around the world, but also in industry because of the potential therapeutic manipulation of this pathway. So you could imagine agonizing this pathway to turn on these antiviral responses. And that's been shown. It's also been shown extensively, and STING agonists are now in clinical development for anti-tumor immunity, where this ability of the STING pathway to drive type 1 interferon is beneficial in the context of anti-tumor immunity, and particularly enhancing checkpoint blockade. So although CGAMP is one way to activate a STING agonist, there have been a number of small molecule agonists described. This is one that was developed by the team at GSK. This is a small molecule agonist that's a very potent inducer of the STING pathway. And we were interested in sort of seeing if we could leverage STING agonists for antiviral immunity. So I'll touch a little bit on that. So these agonists have been published by GSK in that paper I just cited, but just to show you a little bit of the data here. So if you treat macrophages with this GSK 856 molecule over a dose response here, you can see this incredibly robust induction of interferon beta mRNA. This leads to very robust phosphorylation and activation of IRF3. And shown here on the right, it leads to dimerization of STING. So sort of all features of STING activation. So we wanted to see if in this herpes simplex virus model, if we could perhaps, think about a STING agonist as a potential antiviral therapeutic. And we were particularly sort of thinking about this now in the context of SARS coronavirus and strategies to block viral replication. But here's an experiment where we have wild type mice in these black squares. The CGAS knockout mice, which I showed you earlier, were more susceptible to infection than their wild type counterparts. And you can see the CGAS knockout mice succumb to infection. The wild types here at this dose actually are also somewhat susceptible. But a single shot of this STING agonist at 30 minutes before infection completely protected mice from this otherwise lethal challenge. And studies in the lab now are ongoing to really understand, is it all just interferon induction, or is it possible that some of these other pathways that STING elicits are also contributing to this effect? This just highlights some of the weight change in those experiments, and then hydrocephalus here, which is swelling in the brain. You can see the wild type mice in the middle here have some swelling. The CGAS knockout are considerably more susceptible, and you really see no evidence of that after STING agonist treatment. And similarly, looking at a more complex neurological score. So STING agonists basically can protect mice at least from this otherwise lethal challenge. And of course, now we're interested in broader antiviral effects of these agonists, particularly with RNA viruses that might actually manipulate RNA sensing pathways. And perhaps this could be leveraged for antiviral immunity to a broader collection of viruses. So just a slight warning, please do not attempt to take a STING agonist at home, despite these sort of in vivo effects. Okay, so that's kind of the beneficial effects of these pathways. And now I want to move to sort of, the ugly side of these defenses, or the offensive side of immune defenses. So what I've described to you, I think, illustrated in those examples is how engagement of these pathways can be important in host defense. There's a huge literature out there with STING agonists in cancer immunotherapy, potentially in vaccine adjuvants. But for the remainder of the talk now, I want to talk about both my work and also just more broadly put this work in the context of the broader field of sort of where we stand in terms of thinking about how these same pathways can contribute to autoinflammatory and even perhaps to autoimmune diseases. So many years ago, when we had identified AIM2, which is another DNA binding protein that binds double-stranded DNA and forms a caspase-1 activating inflammasome, together with Sam Shao, who's now at Case Western University. Sam had solved the crystal structure of the AIM2 DNA binding domains. And AIM2 binds DNA really independent of sequence. So these sort of positively charged HIN domains are interacting with the negatively charged phosphodiester backbone. And there's no sequence-specific recognition. And similarly, work from many labs has shown that C-gas, like AIM2, also really binds DNA through electrostatic interactions, and it's really sort of, you know, it's not recognizing a particular sequence, it's just merely responding to any DNA. So this highlights the potential for this sort of challenge with maintaining ignorance to self-DNA. So if these DNA binding proteins are merely recognizing DNA through electrostatic interactions, then there's really nothing different about, you know, host DNA versus viral DNA. And you could imagine you could readily sort of tickle these. But there are different ways to sort of prevent that from happening. So there's compartmentalization, where of course DNA is compartmentalized in the nucleus or in mitochondria in normal, healthy, happy cells. And there are scenarios where mitochondrial DNA or even nuclear DNA, you know, is not compartmentalized and starts to gain access to the cytosol, where it could engage C-gas. And now, particularly as we think about, you know, this evidence of C-gas in the nucleus, how do we really sort of prevent C-gas from responding to the huge abundance of DNA within that compartment? And then the other critical mechanism is these waste disposal systems. So shown here is a macrophage. So outside of cells, many cell types in the body secrete nucleases or DNases. So DNase 1 or DNase 1L3, a related nuclease, digests DNA that's outside cells. DNase 1L3 in particular digests DNA from microparticles, or for example, apoptotic cells that have died and have undergone necrosis. You could imagine that these DNAs would normally sort of take care of that DNA and eliminate it from the body. When cells, particularly macrophages, engulf dying apoptotic corpses, another nuclease called DNase 2 in the endolysosomal compartment will clear that DNA and kind of eliminate that threat. And then within the cell, TREX1 is a third sort of class of DNases that eliminates DNAs that accumulate, for example, from endogenous retroelements. So in a normal situation, the kind of combination of these different nucleases prevents aberrant accrual of DNA. But of course, we know that, you know, that does not always happen. So for example, there's clear evidence in both humans and also in mice now that mutations in these DNases outside cells that inactivate the sort of waste disposal function of these enzymes can lead to autoimmunity. And that's depicted over here on the right in this beautiful cell paper from Boris Rees's lab, where they showed that DNase 1L3 really kind of contributes to the clearance of DNA from microparticles and apoptotic cells, and failure to do that as a result of DNase 1L3 deficiency in mice leads to responses to self-DNA and autoimmunity. Similarly, DNase 2, there are mutations in DNase 2 that lead to autoinflammatory and autoimmune diseases, and further support for that comes from studies in the mouse, and I'll show a little bit of a summary of that in the next slide. And then similarly, mutations in TREX1, both in humans as well in mice, lead to accrual of DNA in the cytosol. And then that DNA begins to engage C-gas and other pathways and contribute to autoinflammatory diseases. And then I'm going to come back to SAVI, which is really going to be the focus of the last third or so of the talk. So just to touch on these human diseases. So there's rare diseases, but some patients that have been described that have DNase 2 mutations, resulting from loss of function mutations. And then these patients have essentially a multi-system autoinflammatory syndrome, severe neonatal anemia, kidney inflammation, liver fibrosis, many different sort of organs and tissues impacted, and an increase in anti-DNA antibodies. And even prior to the description of these patients from Yannick Crow in the UK, it was known that DNase 2 deficient mice were embryonic lethal, owing to the fact that there was an excessive production of type 1 interferon in these animals. And you could rescue that lethality by crossing these DNase2 knockout mice with the IFNAR knockout. So mice that couldn't respond to that excessive interferon response were protected from lethality. However, those mice did go on over time to sort of develop a distal polyarthritis. And this was not rescued, you know, even mice that lacked interferon receptors still developed this disease. This was characterized by Nagata's group, and then in a collaborative effort between my lab, Anne Rothstein's lab, and Ellen Gravelisi, a rheumatologist here at UMass, we knocked out either the STING pathway or the AIM2 pathway on this DNase2 IFNAR double knockout background. So now these are triple knockout mice. And you can see if you look at clinical inflammation or the histological score in the joints here, that really the STING knockout completely eliminates disease. And you do also see a contribution of the AIM2 pathway here due to elevated IL-1 that we see in those animals. Just to touch on this third disease, so TREX1, this third nuclease, mutations have been shown to lead to Aicardi-Gouttiere syndrome. This is a disease sort of made famous and characterized by Yannick Crow's lab. Yannick had defined mutations in TREX1 and a number of other nucleic acid sensing enzymes or sensors or proteins involved in nucleic acid metabolism, and defects in these pathways contribute to this disease. So this is a disease that, in contrast to the DNase2 patients, really sort of impacts the CNS. So patients develop severe encephalitis, again, very high levels of interferon, demyelination of motor neurons. And in work from Dan Stetson's group, they showed that mice that lack TREX1, which succumb to a disease in mice that actually turns out to be a myocarditis rather than a neuronal inflammation. But nonetheless, CGAS deficient mice shown here, or sorry, TREX1 deficient mice shown here in black, you know, succumb over time, and you can completely rescue this disease by eliminating CGAS or STING or IRF3 or interferon, and that's beautiful work from the Stetson lab over the years. So from all of this, you can see that, you know, perturbations in the normal functioning of these sort of waste disposal systems can lead to accrual of DNA and start to activate these pathways. And then the most recent disease in this category is a disease called SAVI, and this was described about, I think, 5 or 6 years ago now. It's a disease associated with mutations in the STING protein itself. And again, like many of these other mutations in mice or humans, this results in constitutive activation of these pathways, and patients with these diseases have excessive and sustained production of interferons. So SAVI is the kind of newest, or one of the newest members of these interferonopathies. These are diseases really characterized by high levels of type 1 interferon. SAVI is onset in infancy. It presents with sort of lesions on the nose and cheeks, vasculitis, vascular inflammation, but it's really this sort of interstitial lung disease and fibrosis in the lung that sort of impacts these children in particular. And it's interesting, if you look at all of these different interferonopathies, which can be driven by mutations in different genes involved in nucleic acid sensing, it's particularly intriguing to me that, you know, some of these genes, even though you think the mutation is sort of leading to CGAS STING activation, which is quite a widely expressed pathway. STING is expressed broadly in many tissues. Why you get, you know, different tissues and organs impacted. So many impact the neuronal system, the skin, but the SAVI in particular seems to really sort of be somewhat unique in driving lung inflammation and lung fibrosis. And that's just shown here. So this sort of autoinflammatory interstitial lung disease is really a feature of SAVI that perhaps distinguishes this disease from some of the other type 1 interferonopathies. So the SAVI mutations are thought to mimic a constitutively active STING molecule. So in work from James Chen's lab a couple of years ago using cryo-EM, they demonstrated that a key sort of event involved in STING activation was this conformational change leading to a rotation of the STING C-terminal domain. So when STING is inactive, these transmembrane regions, because STING is a multi-membrane spanning protein, these transmembrane regions are sort of locked and immobile, and these conformational changes really sort of impact the localization of these transmembrane regions, and then this translates into these conformational changes. And the SAVI mutations, there have been many described in patients, are really in this kind of dimerization domain. So the idea is that unlike a healthy cell where you need DNA and CGAMP to drive STING activation, in these patients, STING is constitutively active and is sort of firing down these pathways. So you have active, sustained signaling. And then as shown here on the right, this really leads to these very high levels of interferon in these patients. So compared to healthy controls here, these are SAVI patients, these are TREX1 patients and DNase2 deficient patients. And this is data that was shared with us by Yannick Rowe and his group. So we were interested in trying to better understand SAVI and in particular, understand why the lung might be impacted in particular. And so a graduate student in my lab who's now a PhD, she just graduated last weekend, Mona Matwani, set out to use CRISPR to generate mice that have these mutations. So shown here on the top there, this is looking at the human STING alleles, and on the right-hand side, this is the mouse STING alleles, the sort of corresponding mutations. So we made mice that have N153S, shown in blue, and V154M, shown in red there. And you can see, particularly with the murine STING alleles, unlike the wild-type STING, in a reporter-based assay, so this is just transfecting these alleles into HEC293 cells, you know, all of these alleles are active compared to the wild type, but the V seems to be really more active than the N. And we think this is related to sort of longer term stabilization of this protein. So the V allele is just, as a protein, is much more stable than the N allele. So we made individual mice with the N mutation and the V mutation, and just shown a picture here. You can see that these mice are smaller than their wild type counterparts. And I will add, this is a really close collaboration with my lab and my wonderful colleague, Anne Rothstein, and Kevin Gao is a new student who's taken over this project. So when STING is activated, as I've described, you normally in a healthy cell, you elicit, these interferon responses that can have host defense properties and this anti-tumor property. But in the context of this gain of function constitutively active STING mutant, this pathway was really sort of constitutively active. And to summarize in an illustration here, some of the key features that we found. So fortunately, we were able to find that these mice actually recapitulate many of the features of human disease. They develop splenomegaly, they have thymic atrophy, they have anemia, they develop pretty profound lung inflammation and fibrosis, which I'll show you in a moment. Cells, so there's age-dependent loss of B cells and a pretty profound T cell loss early in these animals. And then there's an expansion of neutrophils. And then as seen in human patients that have this sort of lacy interstitial lung disease, the SAVI mice also go on to develop lung inflammation and fibrosis. So this just shows here. So here again, we're comparing the N to the V mutant, and you can see the V mutants are always a little bit more severe than the N mutant. So these are two of the most common SAVI alleles in humans, and then taking the corresponding mutation in the mouse, you can see in the survival curve that the V mice sort of succumb a little bit earlier than the N mutant, and then looking again at splenomegaly. So both of these animals have increased spleen size. And as I mentioned, they both develop inflammation in the lung, so shown here with H&E staining. So the N mutant and the V mutant on the right. And then when we look at trichrome staining to look at collagen deposition in the lung, we really don't see much of that at all in the N mutant, which is a milder mouse phenotype in general across the board, but the V mice really sort of found age-dependent fibrotic response, and I'll detail that in more details in the coming slides. So the obvious first question that we were interested in addressing was, is this an interferon-driven phenotype? All of these various sort of immune cell and tissue abnormalities are the interferon response. And these mice do have an elevated interferon signature, but it's not a screaming hot interferon signature, unlike some of the other mice for DNase II, for example. So what we found, just very briefly, we took the SAVI mice and we knocked out the interferon alpha beta receptor, or IRF3, a key driver of the type 1 interferon response downstream of the STING pathway. And really this had no impact whatsoever on disease. And that's just shown here with a couple of parameters. So we're comparing now the green bars to the red bars. So red are the SAVI mice on an IFNAR het background. And then when IFNAR is deficient in this context, really no difference whatsoever in spleen weight, in thymus weight, or in the mortality of these animals. And that was true for many of the innate pathways downstream of STING. So we also knocked out the inflammasome pathway by knocking out gasdermin D. It had no effect, or the program necrosis, cell death pathway, similarly had no effect. So we still don't fully understand, at least on a cellular basis, what the downstream consequences are of this response. And then I'll just quickly sort of move through this. So interstitial lung disease is an incredibly important clinical problem. There's really no treatments currently for these, or there are some treatments, but just not very effective for these diseases. And these are quite prevalent diseases. So interstitial lung disease is frequently associated with other connective tissue diseases, for example, systemic sclerosis, rheumatoid arthritis, vasculitis, and also now SAVI. But there are many other sort of forms of interstitial lung disease that are really of unknown cause. The first line treatment for these patients is sort of systemic glucocorticoids or other immunosuppressive therapies, and the SAVI patients in particular have refractory disease. So there's a clear need to understand this process. So the pathogenesis of interstitial lung disease is somewhat poorly understood. It's been proposed that sort of repetitive injury to airway epithelia can damage the airway, lead to inflammation and recruitment of immune cells to the lung, and the expansion of fibroblasts and scarring of the interstitium that would lead to impaired gas exchange. So there's a clear need to better understand this pathway. So we were interested in the possibility that these SAVI mice could provide an opportunity to really sort of begin to, at least in that one context, to understand some of the immune dysregulation that might contribute to this disease. So it's worth mentioning that, as I mentioned earlier, STING is quite a broadly expressed protein. So it's particularly intriguing why gain-of-function mutations in SAVI manifest as lung inflammation as opposed to, inflammation in other organs. And we do see inflammation in other organs, and we are investigating that in these animals, but really it's the sort of lung fibrosis that we think is probably contributing to the mortality of these animals. So STING is very highly expressed in the lung compared to many other tissues. So shown here, this is just looking at RNA data from publicly available data sets. There's been a couple of recent single cell RNA sequencing data sets that have kind of come online, and Jeffrey Zhu, an MD PhD student in my lab, has analyzed that data to really sort of interrogate, you know, in the lung, which cells is STING primarily expressed in. So you can see here over on the left-hand side, in the lung, whole lung, STING is really highly expressed in lung epithelium, particularly in these ciliated epithelial cells, as well as in macrophages. And then in peripheral blood, PBMCs, of course, we know that STING is highly expressed in myeloid cells and dendritic cells, but it's also expressed at very high levels, often higher levels in T cells in particular, and in this particular case in Tregs. And then this is some data from the Human Protein Atlas, really sort of looking at protein expression of STING. And you can really see this beautiful staining in the pseudified stratified columnar epithelial cells that really line the airways, and also in these alveolar pneumocytes. So this sort of expression of STING could really indicate that lung autonomous STING signaling could be the sort of initiating factor that drives lung inflammation and fibrosis in these animals. And we're really actively kind of pursuing this and trying to understand the role of lung parenchyma in this model. And then further, we know that macrophages and potentially other immune cells are dysregulated. So, you know, could they also contribute, or, you know, which comes first, which is the sort of driver of this disease? So we have found that there is BALT formation in the SAVI lung. So bronchus-associated lymphoid tissue is often seen in connective tissue diseases associated with interstitial lung disease. This is just an example of a Sjogren's salivary gland. And this has been seen in the lungs of patients with systemic sclerosis and also some patients with RA. In the SAVI lung, there's sort of clear evidence of BALT formation. So shown here with H&E staining, here with anti-CD20 staining, and then the sort of surrounding CD3 staining for a T-cell rich zone. So are these mice a good model for interstitial lung disease? So do we see these sort of features that are seen in the human disease? So this just shows with a whole lung sort of staining. This is looking at H&E staining or collagen deposition with trichrome staining, and you can see that the VM lung also develops BALTs and interstitial lung disease, kind of similar to what's been seen in human patients. And we wanted to understand if there were B cells and T cells accumulating in the lungs of these VM mice. So to do that, we used this anti-CD45.2 antibody staining procedure that allows you to distinguish intravascular from extravascular immune cells. So the extravascular cells would be in the alveolar space, and then you could determine, you know, are the cells in the lung in the intravascular or extravascular space. And shown here on the right, you can see for B cell staining that there's both the number and the percentage of B cells in the extravascular space in the lung is elevated in these animals. And we see similar things with CD3 positive T cells. So there are both B and T cells in the lung. And then we took the SAVI mice and we crossed them to RAG deficient mice, and we could completely rescue these immune abnormalities. So the splenomegaly on the left here, and then you can clearly see that the RAG knockouts really eliminate these BALT formation with H&E staining, and collagen deposition is completely reversed. And this really required RAG deficiency because TCR beta deficiency was not sufficient to alter these sort of readouts of disease in these particular contexts. And just briefly, the T cells in the lung are producing interferon gamma, but if we cross the SAVI mice to the gamma receptor knockout, similarly, we don't lose inflammation or fibrosis in these animals. So this clearly suggests a role for lymphocytes. And I want to mention similar work was published, actually our work on the RAG is still unpublished, but similar work was published by Jonathan Miner's lab at WashU using the N153S mice, where they actually found, so this is a milder disease that develops inflammation, but not really, at least not in our hands, not fibrosis. And deleting T cells really limited disease progression, and RAG deficiency, much like I showed you with the more severe V mouse, also eliminated disease. So just like in the human context, then, you know, we think this mouse model of SAVI, you know, is a relevant model to study the interstitial lung disease in these patients. And just lastly, looking a little bit more at B cells. So we're obviously focused now on the role of B cells in this disease. If we look at the B cells in the lungs of these animals based on CD69 and CD86 staining, these B cells are highly activated. The activation of the B cells seems to be independent of alpha beta T cells based on our analysis in those mice. But of course, you know, these animals, their lungs are filled with gamma delta T cells, and we're in the process of interrogating the role and contribution of gamma delta T cells. Just in the interest of time, I'm going to skip through this, but we've been profiling those B cells, and in the extravascular space, there's clearly an accumulation of B cells, and these B cells are highly activated based on MHC class II expression. So our attention now is really focused on the role of B cells in the SAVI fibrosis model, and B cells certainly have shown some initial promise in interstitial lung disease in patients. There's been a couple of studies looking at rituximab that showed some benefit, but you know, through this SAVI mouse model, we're hoping that, you know, we can use this as a model to really sort of interrogate, you know, the contribution not only of B cells, but also the lung parenchyma, particularly these lung epithelial cells, and looking at both hematopoietic and non-hematopoietic cells and how they contribute to initiation and development of disease. And really, there have not been good models of interstitial lung disease, so we think this SAVI model represents such a model. So just to wrap up, I know it's close to the time here, so all of this certainly suggests that, you know, antagonizing the STING pathway could have benefit in a disease like SAVI or some of these other diseases that I have described. And there's a large effort now in pharma and biotech to develop small molecule inhibitors for these pathways. Many of the small molecule inhibitors that are in development are sort of CGAMP competitive inhibitors. So I think in the case of SAVI, you know, targeting a different aspect of STING function will likely be required, since this is really STING activation independent of CGAMP binding. But in some of these other interferonopathies, CGAMP competitive inhibitors would likely show promise. All right, so I'm just going to skip that because I've sort of gone through that already. So just want to acknowledge the work. So as I mentioned, Mona was a grad student who started this work. This is now work with Kevin Gao, an MD PhD student. And all of this is really a very close and productive collaboration with Anne Rothstein's lab. We actually co-mentor both of these students together. And then other people in my lab who are looking at different aspects of SAVI inflammation in vivo, Laraz and Fitzgerald Humphreys. I want to acknowledge some of Anne Rothstein's lab who have contributed in various ways to the SAVI project. And then some of the other people shown here for some of the help with some of the flow experiments. And then just lastly, in my own lab, I talked a little bit at the start about some of the herpes simplex virus work, and and the STING agonists, and really Fiachra Humphreys is a driver of all of that work. And that involves collaborations with Evelyn Kirk-Jones, and I mentioned some of the collaborative work with Ellen Grevelisi. And we're grateful to all of our funders and collaborators and other members of my lab and previous members. And just to mention one last thing, for those of you who are enjoying these sort of Zoom type seminars, there's a new series that actually is about to start June 22nd. This is developed by Claire Bryant at the University of Cambridge, and it's really a Zoom seminar series that's focused on inflammasomes. So if you love inflammasomes, this will be your home. Okay, and I think I'll wrap up there.
Thank you so much, Kate, for such a wonderful talk. Thanks for sharing this information of the inflammasome.
Yeah.
That's really cool. And thanks for sharing that really unexpected role of B cells. I think they're coming to the forefront of so many.
I know. Who would have thought I'd be working on B cells? But you know, it's good to have Anne Rothstein next door.
Exactly.
It's my B cell queen.
Yeah, absolutely. Thank you so much. And so before we finish.
Thank you, Kate. That was beautiful. Thank you so much.
Thank you. Yeah, happy to do it.
Yeah. So before we finish, we just want to remind everybody that Kate will be able to answer questions via Twitter. So if you search for Global IMMUNOTALKS, you can find the tweet that hopefully will be up very soon. Sorry, that says ask questions for Dr. Kate Fitzgerald seminar here. And Kate uses Twitter, so please just mention the hashtag GlobalImmuno and add Fitzgerald Kate, and she will be able to address your questions. And if you are listening to this maybe later on, maybe tomorrow in YouTube, we will be monitoring and trying to answer questions later as well. And just a reminder, hopefully you can join us next week. We have a little bit of a change in our schedule, and next week we will have John Werry joining us as a global immuno speaker. So thanks everybody for joining across the world, and we hope to see you next week. Thank you, Kate.
Bye.
Thank you. Thanks so much for doing this. Bye.
Bye. Thank you.